Let's understand physiology of distal tubule and collecting duct. There are two parts of distal convoluted tubule, early and late part. Early part of distal tubule is impermeable to water. At the apical membrane, sodium chloride pump and calcium channel is present, while at the basolateral membrane, sodium-potassium ATPase pump, calcium ATPase transporter, sodium-calcium transporter and PTH1 receptor for parathyroid hormone is present. Let's see how absorption occur here. Filtrate coming from ascending limb contains various solutes and water. At basolateral membrane, sodium-potassium ATPase transporter pump sodium out and potassium inside cell, decreasing sodium level inside cell. Sodium present in filtrate will move toward low concentration in cell and also bring chloride ions with it. Chloride ions will move out of the cell to blood through special channels. Absorption of calcium is regulated by parathyroid hormone. When there is low calcium level in blood, parathyroid gland release parathyroid hormone in blood, which will bind to PTH1 receptor. This activate GS-GTP complex. This further activate adenyl cyclase. Adenyl cyclase convert ATP into cyclic AMP, CAMP activate protein kinases A, which activate calcium channel by phosphorylation. Now calcium will be absorbed from filtrate through these channels. Calcium bind to calbindin protein inside cell and will be transported to blood through sodium-calcium antiporter channel and calcium ATPase channel. Here energy is required for the calcium to go into blood because calcium in blood is still in high concentration as compared to cell. Next is the late part of distal tubule along with collecting duct. There are two parts of collecting duct, cortical and medullary collecting duct. In physiology, late distal tubule and cortical collecting duct are grouped as the aldosterone-sensitive distal nephron, although anatomically they are distinct segments. Both have similar cellular functions and aldosterone sensitivity. They contain principal cells and intercalated cells. Principal cells maintain mineral balance and water absorption. Intercalated have two types, intercalated A and B cells. These cells maintain acid-base balance. Let's first see principal cells here. At basolateral membrane, sodium-potassium ATPase transporter pump sodium out and potassium inside cell, decreasing sodium level inside cell. Sodium present in filtrate will move toward low concentration in cell and excessive potassium inside cell will leave into the lumen through potassium channel. Reabsorption of sodium is regulated by aldosterone hormone. Stimulus that release aldosterone hormone include angiotensin due to low blood pressure, when sodium level in blood is low, and when potassium is high in blood. Aldosterone coming from adrenal gland, as it is a steroid hormone, it diffuses through the cell phospholipid bilayer. It increases activity of sodium-potassium ATP pump, and bind to mineralocorticoid receptors on nucleus. Activate transcription factor that increases production of epithelial sodium channel protein which will be displayed on apical membrane, increasing sodium reabsorption and potassium excretion in filtrate, or we can call it urine. Here 4 to 5% sodium is absorbed while 5 to 6% sodium is absorbed in early part of distal tubule. Now comes the water reabsorption. Normally this area is impermeable to water completely, but ADH hormone make it permeable. So, how does the antidiuretic hormone come and play its role? Its production can be stimulated by the release of angiotensin II in response to low blood pressure, and when sodium level is high in blood due to dehydration, meaning high osmolality of blood. So, the antidiuretic hormone released from the pituitary gland in response to one of the stimulus, binds to V2 receptor present on the cell at the basolateral membrane. This activate adenyl cyclase via the GS-GTP complex. Now adenyl cyclase convert ATP to cyclic AMP. CAMP activate protein kinases A. Now, there is a vesicle containing aquaporin 2-channel proteins. Protein kinases A phosphorylate these AQP2 proteins in vesicle, signaling them to fuse with the apical membrane of cell. Now water is reabsorbed from the lumen via AQP2 channels into the cell following sodium. Water will pass to blood through aquaporin 3 and aquaporin 4 channel at the basolateral membrane of the cell. Here is the small reminder, aquaporin 1 is present in descending limb of loop of Henle. Next are the intercalated cells. Intercalated A cell play role in acidic condition like metabolic acidosis and respiratory acidosis. In such condition, pH of blood decrease due to increased level of hydrogen ions. Let's see how it happens. When carbon dioxide level is high in blood, carbon dioxide diffuses in this cell, combine with water, forming carbonic acid. This breakdown into hydrogen ions and bicarbonate ions. Hydrogen ion will be secreted into the lumen through hydrogen ATPase channel and hydrogen potassium ATPase channel in exchange of potassium ion, while excessive potassium will leave the cell. However, in proximal tubule, hydrogen was secreted through sodium antiport. On the other hand, bicarbonate will be transported to blood through channel at the basolateral membrane in exchange of chloride ions. Bicarbonate ions will increase the pH. Intercalated B cells maintain pH in basic conditions like metabolic alkalosis and respiratory alkalosis, when pH increases due to low level of hydrogen ion. Here, the mechanism is opposite to the above occurring in intercalated A cells. Hydrogen ion will be transported to the blood instead of lumen, while bicarbonate will be transported into lumen instead of blood, through channel at the apical membrane in exchange of chloride ions. Here hydrogen ions will neutralize the basic pH. Potassium and chloride will be absorbed into blood. Aldosterone also have receptor on intercalated cells. On type A cells it increases the activity of the apical hydrogen ATPase pump and hydrogen potassium pump, promoting the secretion of protons into the urine to excrete acid, and increase the bicarbonate chloride exchange at the basolateral membrane. Physiology of medullary collecting duct is similar in a way that it absorbs sodium through principal cells regulated by aldosterone, absorb water regulated by ADH hormone, maintain acid-base balance. However, it absorb urea with the help of ADH through UTA1 receptor and transport it to medullary space through UTA3 receptor, where it maintain medullary interstitial hypertonicity.